organism, and I'll hear like strepto, it's like that, no, that's not the shape, that is the arrangement. There are only 3 shapes, cocci, bacilli, spiral, that's it. And then how do they hang out together is single, diplo, strepto, and if you're a cocci, staphylo, okay? For the bacilli, we have singles, diplos, streptos, no staphylos. There are some that are kind of not quite a cocci and not quite a bacillus. This is the coccobacillus. And in fact, Streptococcus thermophilus that we looked at many times in the lab, that's the other one next to lactobacillus that makes yogurt, it's more like a coccobacillus, more like an oval. Okay, so we have spiral that we know exist, and that's, we're not going to be able to look at these in lab. All the ones that are out there are just a little bit too close to being pathogens that we can't really play with this in that class. Every single spiral organism is flagellated, every single one. So they are always singles. Rarer shapes, not something I'm going into. Skip slide 6. Okay, so starting from slide 7 on, it's basically looking at an actual organism on the right, and this is what kind of electron microscopy is it? Well, the T, there's a T.
Transmission.
So it's transmission. And so if you see an image and it looks three-dimensional, that's scanning. So a scanning electron micrograph, like this one, SEM, you know, okay, I can show that to you here, but if I print it, we do not have a color printer. It's black and white, so I may or may not show something like this. I will if I can find something that looks three-dimensional despite being black and white. Okay? We just have to kind of recognize that we're not doing it based on color. We're basing it, does it look three-dimensional? Okay, the scanning electron microscope is taking like thin slice picture, and then like compressing it to form one image. Okay? All right, so then looking from the outside in, we started with, well, first off, we note that all 4 structures that I would find in any given cell are in fact present in bacteria. So we have the plasma membrane, we have the DNA, we have ribosomes, we have a cytoplasm. All cells have this. These are 4 structures. So remember, we have the elements, we have the carbon compounds, then the structures, right? Okay. And then working from the outside in capsule, an encapsulated organism, we note that any organism that has one is virulent, and that it extends far beyond the cell itself, and it is composed of a polysaccharide, and so it takes on a lot of water, much like a cracker would if you were to drop it in water and come back later. It would be slimy, and because it's slimy, then my immune cells cannot grab it. It also ends up being very important in healthcare because it's resistant to antibiotics, to antibodies. The phagocytic cells can't grab it. Very hard to clean. Okay? And so because it starts on a non-living surface, it's going to be some artificial material that was introduced into the person. So the catheter is the #1 nosocomial infection, is a UTI, the urinary tract infection. That is 40% of all nosocomial infections, but not far behind are those caused by ventilators that actually have encapsulated organisms in the tubing, and then IV lines, but it's also hip replacements, knee replacements, stents. Just think of some artificial thing that has been put into a person that is a potential site of this happening, okay? So, and we do a lot of that, and we have an aging population, so this is just routine for us now. I mean, you like name somebody over the age of like 65, and there's a very high likelihood they either have a knee replacement or they're heading for a new one, right? This is kind of that one. So we just keep that in mind. If an organism can move, a bacteria can move, then we know it has only one structure for that, and that's flagella. So how the flagella arranges is not something I can show you. Today in lab, we do something called the hanging drop that allows us to detect motion. We call it true motility. If we see it, we know the organism is flagellated, but I will not know in what manner. I won't know if it's paratrichous, I don't know if it's lophotrichous, I'm not gonna know that part. Okay, that's, for this, I need the scanning electron microscope. So I can't look at that. Okay. Flagella occur in both gram-positives and negatives. Okay, despite the fact that we keep looking at gram-negative flagellated organisms in the lab, and so then you start thinking that only gram-negatives are flagellated. That is not the case. Okay, so it occurs in either one. We are skipping slide 27, slide 28, 29, so that's like just, it's too detailed, and it's not anything I can show you. Okay, fimbriae, however, are these sort of long projections that we encounter on gram-negatives only, and they use it as sort of like Velcro to stick. And so this doesn't really mean much to us right now, but it will, because when we get to normal microbiota and how it is that they hang out, then you'll know that this is how, is that they have this fimbriae and they're able to stick it out. Okay, so we can skip 33 for the moment because, you know, we're gonna get to that. That's unit 2 also. And then skip 34. All right, 35, we're on cell wall. We know it's important to keep it from bursting. And then we started talking about what it actually looks like. And so I draw this on the board for you today because we do the gram stain. It's very important that we understand this. So it is composed of glycan rods composed of these alternating sugars, N-acetylglucosamine and N-acetyluramic acid. But you know, chemistry has this like tendency to make us like, I don't know what I'm looking at. It's like, okay, I'm looking at a hexagon. When I see a hexagon, that's a glucose. And then you just put a bunch of weird groups on it, and so it has a different name, but the base structure of it is a glucose. And so what do I know about sugars? Do they like water? Yep. Okay, so it's like the chemistry required in this class is like really minor. You like water, you don't like water. Are you charged, not charged? That's it. So not a lot, and I'm not gonna let myself get freaked out by this. I'm looking at a bunch of sugars. Okay? These glycan rods are cross-linked with amino acids. Remember that I said that which amino acids and how many, that's gonna differ between different bacteria, so it's not worth learning that. Okay, but what we do know is it doesn't matter which bacteria it is, this is what they're doing. They're using amino acids to hold together these glycan rods. So just for a moment, let's pretend it's not peptidoglycan that we're making, not the cell wall, but just like it's a raft. Okay, so you cut down the logs. And if that's it, this is not going to have much structural integrity. You're gonna get on the water and the logs are gonna go away from each other, right? You can see that? So this is the purpose of cross-linking it, and they use amino acids. Amino acids, remember, are linked by a peptide bond, which is very strong. That has partial double bond character, okay? Now, the thing is with bacteria is that the amino acids that they use, they sometimes will use, they always use, some D amino acids. That's in a different enantiomeric formation than the L, okay? Humans only use the L. And so when you get the D introduced, we mount a response because it's wrong to us. Okay, it marks it as not self. So I don't need you to know what is the name of that amino acid or anything like that, just that they're using a different version, like a mirror image, and it's not right to us. Okay? All right, so then, yes?
You keep saying
bacteria? Yes. Yes, sorry. Bacteria use deformation amino acids to do this amino acid cross-linking between the glycan rods. Now, what you may note here is that the cross-linking is with the N-acetylmuronic acids. It's the NAMs doing it. Okay. All right, so then the gram-positive has a very thick layer, okay? And I see 3 here. Okay, how many can I anticipate?
20.
Okay, so inaccurate picture. And in order to hold this wall down, we have these lipoteichoic acids. And I recognize that a black line isn't really making you think of an acid, but this is what they've chosen to use. Right? Lipoteichoic acids, that's going to be a carboxyl group that's going to be negatively charged. We have lipoteichoic and wall-teichoic acids. Which parts of these things can I see in lab? None of them. Okay, just know that they're there because I'm in lecture and I'm being told this, but it's not something I can see in lab. Okay? So a thick layer of cell wall, and then for gram-negatives, they also have peptidoglycan. It's just thin, 1 to 3 layers. Okay? And whether it's 1 layer or 3 layers, each one of these glycan rods must be cross-linked with amino acids. They just haven't bothered to show it to you. Okay, so that would still be the case. Beyond that, though, we have this outer membrane. So the outer membrane here, first off, it's another phospholipid bilayer. So we have phosphate heads, lipid tails, but we also have lipid A as one of the lipids, and let's see where I have that picture. Yes, here. Okay. All right, so this is showing us the lipid A. This is the lipid, okay, and then we have the O polysaccharide right there, and we have foreign proteins which look like this, beta barrels. You know, we have alpha sheets or beta sheets, alpha helices or beta sheets for the secondary structure of proteins, and this is a beta barrel, and so it's a pore that comes through. But between the two of them, the lipid A, excuse me, the LPS, because it's the whole thing that takes part in this, and the porin, they repel anything that's large or hydrophobic, which turns out is a lot of drugs, dyes, antibiotics, and bile, like a bunch of stuff. So, but it does, some outer membrane proteins can evade phagocytosis, but here we have heavy metals can also be sort of repelled. Okay, and then the gram stain is today's lab, so we can skip through that, okay? We do need to know the false negative. I may or may not have the number of questions required in lab to ask you that on the lab exam, but it will be asked here. What is a false negative, and when and why does it occur? So a false negative is a gram positive that looks pink. And the reason is after 24 hours, the thick layers of peptidoglycan start to slough the way you would slough your skin, okay? Shed. And then once it gets thinner, now the ethanol step will in fact be able to rinse away the dye, and now they're going to take up the second dye, the counterstain, safranin, and they'll wind up pink. So it's just something to keep in mind when you take your patient samples and send it off. Just kind of keep track of it and make sure you get your answer before 24-hour period. Otherwise, I'm not sure it's going to be accurate. Okay. There are 2 very important types of bacteria that cannot be gram stained. One is the mycobacteria because they have a 70 carbon long 70, yeah, I know, it's a lot. 70 carbon long lipid layer called mycolic acid. And it's so many carbons and it's so hydrophobic that it's more like a wax than it is an oil. And so because of that, it repels anything that's charged. And so the entire purpose of using crystal violet and sopranin is because they're positively charged and they're attracted to the negative charges on the gram-positive, gram-negative. There's no charges here. It's a big, thick hydrophobic layer, so it can't be stained. And what you have to do is do a negative stain, which means everybody but them gets stained. And you see how they're the clear areas there? Okay, what shape is that? That's bacillus. Yeah. And you know, this is just one of those things, you just have to go over it a bunch of times. And I told you before, this is like learning French. Like, you know, it doesn't, you don't know right now the name, the French word for some obscure item. You have to like have a flashcard and like remind yourself. It's not going to come to you. So don't feel bad if I just don't know this. It's just kind of one more time. That's it. Okay, it's not beyond your ability. Okay. So, but there is no secret way to get good at this. Okay, the secret way is put the time in. Okay, it's not a secret. So, the other type is mycoplasma. And so one thing to notice is mycobacteria sounds a lot like mycoplasma. How are you not going to get them mixed up? You're going to remember that mycoplasma, their outermost layer is the plasma membrane. That's it. They don't have a wall at all. No wall. So if you think, especially morning group that just did the gram stain, the entire premise of why you would be called a gram positive is because, what do we say about gram positives? That they retained the initial stain. And where do they retain it? In their cell wall. There's no wall here. So like, none of this is gonna work. And so here's the bad news about the gram stain. It's super easy. It's not really hard. I mean, it's hard today because you're new at it, but you do it like a bunch of times. I'm sorry how many times you're gonna do it, but you're gonna do it a lot. The bad news about the gram stain is if you will not keep stain 1, you're pink in the end, because at the end it's just a pink wash for everybody. And so you'll see later when we do some more descriptions of certain organisms, like say Treponema pallidum, the causative agent syphilis, they'll say it's a gram negative. It's like, is it? It's just pink at the end. Okay, so the gram stain is really kind of, if you don't have a wall, of some kind, like we don't know what's happening. Okay, so there's limitations. It works for the majority of bacteria. That's why it's used all the time. All right, so I want you to just, like 54 and 55, I really want you to get these straight. And the reason is because I hate to tell you, mycobacteria, there's like a tuberculosis outbreak right now apparently in like, what is it, Kansas City, Missouri? Like, what? It happens. It's highly contagious. And it's not like measles. Not as bad as measles. No. But it is respiratory. We have to breathe. Okay? When something is a fecal oral thing like norovirus, like you can keep yourself safe from that. Wash your hands. Cook your food. You're gonna be fine. When something is in the air, I can't see that something is in the air. And I can hold my breath for only so long. I'm gonna take a breath. That's the way that goes. So these are the tough ones. Okay, so it's not some rando thing I'm trying to make you learn. It's like you're gonna run into this guy and mycoplasma too. Okay, so UTI is a big issue. Walking pneumonia is the other. Walking just means you don't see, this is the bad thing. You don't even feel bad enough to stay home. You're out there spreading it. Okay. All right, so then we move down to plasma membrane. We've had a lot of plasma membrane conversations, so I feel good about that. So phosphate heads, in and out, tails in between, works great at homeostasis. We have embedded proteins to do all the work. Okay, anything that's small and hydrophobic is going to come right across. We're skipping 62. Okay, 63. Selective permeability. Okay, so yeah, stuff, if anything is large or charged or, I've got too much already, I'm just going to use a protein to get in there. Okay, that's fine. But here's something that they will do, and this is unique to them. So remember, we have like a whole thought process of like, this is only true for bacteria, this is only true for eukaryotes, this is true for both, and this is true for neither, for example. You throw this on there too. Okay, group translocation. And so once they bring material across, they can trap it inside with the addition of a phosphate. They can add other groups too, but phosphate's very typical. And by adding a charge, it's not going to leak across this phospholipid bilayer because the tails will repel it right back inside. Does that make sense? Okay, so honestly, if you understand polarity, you're like 90% of the way to what you need to know for this class. Okay? So that's it. And it's, you know, it's really helpful in like real life too. Like you're washing your hand, this thing will not come off. It won't come off with water. Go put some oil on it. Watch it just spill off. Because that's it. That's all there is, thankfully, in the world. Polar or no. That's it. Okay? And I will know my answer instantaneously whether does water like it. If water likes it, that thing's polar. That's all I need to know. Okay? Non-polar, just get some oil. Whatever oil you're willing to sacrifice. Okay? So we know these words: isotonic, hypotonic, hypertonic. All right, and so the cytoplasm is mostly water, but it does have dissolved proteins, carbohydrates, lipids, this kind of thing. We have DNA. It's one circular piece. It's a double-stranded helix. When they divide through binary fission. Very, very simple. They will copy their DNA, and then they extend their wall, which means they're going to need to make more of everything, more plasma membrane, more cell wall. So this is very anabolic, a lot of building, a lot of energy required to divide. And remember that lipid synthesis happens in the plasma membrane. Remember that part? Okay. They also do ATP synthesis. Okay. Their ribosomes are a little bit smaller than ours. Okay, a little bit. Okay. We're skipping 72, 73, and now 74. Are we here? Yep. Did I not spend enough time on ribosomes, or are we good? It's just, they're just a little smaller than ours, that's it. And when that's the case, whenever we have structures that differ between our cells and bacterial cells, and here I was going to say they again here, okay, then I can aim at that. And so I have actually a whole class of antibiotics designed to destroy bacterial ribosomes. This isn't really something we need to know now, because I have an antibiotics lecture. It'll take me 2 times to get through, and we have a lab in it too, so it's like we learn antibiotics. So this is just a kind of a thing to name. Inclusions, there's a certain ways to stain for particular inclusions, and that would help you determine which bacterium you're looking at, but we're not going to do that this semester, so we're just going to skip that. All right, but here, endospores, very important. Very, very, very important. Okay, because what is the definition of sterile? Including endospores. Here they are. So I told you, just write it down, and we will get there. But I'm telling you about it now. So first of all, the organisms capable of sporulation, of forming an endospore, are not the majority of bacteria. They're a minority. And those are typically gram-positive bacilli found in the soil. And so maybe right now you're remembering, oh yeah, I measured out across the ground of dirt. And I made pollutions. And I put them on a plate. Because we're trying to find some of these exact organisms. Okay, and black, and I told you it takes forever. So you just record it, so you have to let things work. All right, so gram-positive bacilli from the soil. Those are the typical sporulating organisms. When those organisms find themselves in a situation where there's no nitrogen and no carbon, then they will begin sporulation. There's other things that would bother them enough to start sporulation as well. Antibiotic use. Okay, would be another. No nitrogen, no carbon means they can't build the 4 carbon compounds. What are they?
Polysaccharides, lipids, proteins, nucleic acids.
That's right. And the carbon's in everything. The nitrogen, we would find that for them, you know, NAG, they got nitrogens in their cell wall. They're also going to have that in amino acids, okay, nucleic acids. So basically, they can't build their stuff. And if they can't build their stuff, they can't divide, right? So they're going to undergo sporulation, which is a resting state. It will be a lot like the way a seed is a resting state, but a seed has nothing on an endospore. Okay? So sporulation is not a form of reproduction. It is instead a form of hiding, of resting until things get better. Okay? So we want to know who's likely to do it and under what conditions, and now I'll tell you the how. Okay? So what they will do, if this is the case, out of nitrogen, out of carbon, then they'll do this. They will in fact copy their DNA. And we know that the DNA is held at one point at the plasma membrane, that's one of the functions of the plasma membrane for bacteria. So they copy their DNA, and typically if this were binary fission, they would just split down the middle, okay, as I showed you a moment ago. But this isn't reproduction. So what happens is they start pulling apart the cell, but instead of splitting down the middle, notice that I have a big side and a small side. The big side is called the sporangium. The small side is called the forespore. Okay, so they split. And you notice this yellow line here, that's the plasma membrane, right? And that's the plasma membrane. So this plasma membrane that was contiguous here has now split, okay? And this sporangium is going to swallow the forespore. Now, I am trying to describe something that isn't static, but rather an active process. And so it's not a bad idea to go on YouTube and just say like endospore formation or sporulation, and then do this, plus kids. Did I tell you this already?
No.
Okay, so I had a student that got a D in this class. And I saw her like 3 semesters later and she got an A. So like, let's talk. And there were a couple things. She had to like set some things in her life aside and make time for the class. She realized how much time was required. But the other thing she told me is that she had this little trick. When she didn't understand anything, she would go on YouTube, type in the search bar, whatever the concept was, and then the Boolean operator plus, and then kids. Something that was designed to teach like a 7th grader. And when she understood that, then she took the kids away. And that's how she did it. So I keep in close touch with former students, right? It's like, bring it, right? Because at this point, every year that passes is another year that I'm further and further away from what it is that I did, right? Which may or may not even be really that relevant to you, okay? So, same thing. If you're finding some really wonderful thing, you know, don't keep it to yourself, bring it. All right, anyway.
What did you say to look up for this?
Sporulation.
Sporulation.
And then I put the plus kids, you know. And if it goes into more detail than I'm talking about, then just skip over that part. Okay? All right, so they copy their DNA, they split the cell, but not in the middle. It's sort of one small area and a larger area. The larger area swallows the small. So now this early spore actually has 2 phospholipid bilayers. One that it had on itself right here, and then this one that engulfed it, okay? 2. And between those phospholipid bilayers, the plasma membrane, it deposits a lot of cell wall. So remember, the cell wall is the glycan rods crossing with the amino acids. Then this cell starts to produce a lot of fibrous proteins. Proteins come in 2 forms. Globular, those are soluble. Fibrous, those precipitate. Hair, is keratin. That is a fibrous protein. It's insoluble. If you showered, you still have your hair, didn't dissolve, okay? Fibrous. So they start filling up with fibrous proteins, and that forms this cortex right here. Cortex. Okay? And so they are now protecting the DNA with this cortex. But the other thing they do is produce something called dipolcolinic acid. That's just right, DPA. And they deposit that all around their DNA. So now their DNA has two things protecting it, the DPA and the fibrous proteins. And the other thing that they will do is they start pumping out water. So all the water from the cell, and remember, cytoplasm is what percent water would you say? Yeah, it can be up to 80, like 70 to 80% water. They just pump it out. Okay? And so once that is done, the normal cell just gets destroyed, but really that doesn't even matter because this thing gets released. And so now you're looking at the spore, and this spore is very resistant. So it has, because of the fibrous layer protecting the DNA and the dipolcolinic acid and the fact that it's gotten rid of the water, it can withstand many, many things that would kill an actively growing and dividing cell, which is called vegetative. So let me write that down. Vegetative bacteria are actively growing and dividing. And what we're talking about here is an endospore. This thing is a resting state. And so, typically, if you boil water, okay, then you're raising the temperature. And you know, temperature is just a measure of molecular motion. So, okay, all the molecules are moving really fast, right? And so the water on the outside and even on the inside of the cytoplasm starts to move so fast that this would kill that cell. You can boil water and kill off the bacteria in it. Boiling does not work on an endospore. Why? There's no water in it. That's part of sporulation. They got rid of the water. They took care of that part. Okay? I can destroy bacteria by exposing them to sunlight. It totally works. UV will work its way through and it will hurt their DNA and it will kill them. UV is not strong enough to penetrate the spore coat. UV doesn't work on them. I would have to use an X-ray. Okay, that's nice to know, and you can write that down. Who the heck has an X-ray? Like, you don't have that. So, I mean, those of you going into dental hygiene, okay, you want access to it. Do not play the X-ray. But okay, so that's one thing. And then a bunch of chemical agents that you would use to destroy bacteria, and it would totally work. You know, bleach, right? All kinds of cleaning agents. It doesn't work. They have found spores in liquid amber, okay, so rocks, and the estimate is they're like over 10,000 years old, and they can get them to germinate, which means like kind of spring back to life. Okay, so they're tricky, and if I want to get rid of them, then I have to sterilize. And that is a word that I use sparingly. And if something is sterile, I'm saying that everything is gone, including these guys. So, your sister just had the baby. And she says, I just sterilized the baby's spots. And you're like, what'd you do? Like, you're not going to break her heart. Okay. Okay, we're not going to say that. Okay, that's unit 2, and we'll learn all these words, but I assure you that all these manufacturers of chemicals, they know these words. If they say sterile, that works to sterilize. If they say disinfect, that's what that does. If it says sanitize, that's what that does. These are different words. They have different layers and levels. So I don't bandy about the word sterile or sterilize. Yes?
What exactly does it take to get through those layers?
So we're gonna sterilize, right? So for example, holding something metal in a flame until it glows red is enough to sterilize. Okay? Putting something in an X-ray machine is enough to sterilize. There's only certain things that can be done. The other thing is an autoclave, and that's what happens to all of our materials in the lab. An autoclave is sort of a cross, bless you. It's a cross between an oven and a pressure cooker. So it raises the temperature a lot and puts it under 15 pounds of pressure. We're experiencing 1 right now, so 15 pounds of pressure is enough to like burst through that spore coat. But you know, who has an autoclave? That's the labs do, okay? Here, the things that I hand over to you have been autoclaved, but otherwise, you can buy something that's sterile. It was made that way in the manufacturer, but now, if I can just like glance at it and it's so beautiful, but I have to use it. So to open it, I have to do the aseptic technique to minimize the chance of contamination. Once it's open, it's over, right? We know this. So this is sporulation. I do recommend looking online. If I can find one between now and Wednesday, I'll see if I can, give my tacit approval to this. But YouTube will have, thousands, and I'm not gonna be able to watch them all. I'll probably look at 1 or 2 that look okay. If it goes into like super detail, I didn't give you super detail. I'm not expecting super detail from you. I want you to know that this thing is a resting state. It's highly resistant, and to kill it, we have to sterilize. Okay, and that's knowing how to sterilize and all that stuff. That's unit 2. We just want to know what it is that we're after. So I define. It's a little bit off because I define sterile to you like day 1 of this class, and you're just like writing it down. I was like, just write endospore, we'll get there. And now we're there. Okay, you had a question, Palmer?
Oh, if they're not vegetative, then and they're not like growing and dividing, they're still bad though. Even
Like
though
what?
They're not
Like endospores?
Yeah.
Okay, so here's what happens, because I haven't talked about germination quite yet. When the endospore finds itself in an area that is moist and has lots of nitrogen and carbon, let's think, what might that be? So you might remember that when we talked about Dr. Cope, and he was working with anthrax. Okay, anthrax, causative agent, Bacillus anthracis, causative agent of anthrax. Bacillus anthracis lives in the soil. It's a gram-positive bacilli. Okay, that was used as a bioterrorism agent right after 9/11. So in 2001. I mean, this had nothing to do with the towers, but everybody was just already freaked out, and somebody, I don't even know if they really figured out who was sending out envelopes full of anthrax spores. This was like a white powder. And he sent it to this guy, Tom Brokaw, who was at the time the anchor for like NBC Nightly News, so at the Rockefeller Center in New York City, addressed to him. He doesn't open his own mail. So his secretary opened it up, all his powder came out, she breathed it into her lungs, which are warm, moist, and full of nitrogen and carbon. So they allow moisture back in when they get into a moist environment, moisture back in, and now, sorry. Okay, so moisture will come back in, and then enzymes within start to break apart the fibrous proteins, and now it becomes vegetative all over again. And now they're actively growing and dividing. Unfortunately, she passed away. But as soon as, and they figured out immediately that that's what it was, because it was like she started coughing, and the next day, like her lungs are like bleeding, and like she's dead. And so they just put the entire floor, and if you've ever been to Rockefeller Center in New York, that's huge, way bigger than this building. So like the entire floor was immediately put on ciprofloxacin, which is a type of antibiotic, which is like the big guns. Right? Because, and no one else does. But spores can be dangerous. They can be bioterrorism. I mean, that's like a one-off. That's not like life. In real life, what's happening is things like this. Oh, I can't tell you because I move forward. Here's who here's who we need to think about in healthcare, okay? Here's who we need to think about. Clostridium. Clostridium are a type of soil bacteria. They are gram-positive bacilli. There's several different kinds. There's Clostridium Clostridium tetani. Guess what that causes? Tetanus. Botulinum. Botulinum. Yes. Okay. Perfringens. This one causes gas gangrene. And this one you think, I don't need to worry about that. Yes, you do. Because it used to be, are you trying to ask me a question? Oh, I'm sorry, I couldn't tell. You're like, just holding your head up. Okay.
I was gonna ask, is it possible for an endospore to survive sterilization?
No.
No. So
No, that's the definition of sterilization.
it's just like, no matter what.
Yeah, so okay, so perfringens causes gas gangrene, and it used to be like, you know, if you worked in like an army hospital, okay, that's who has to worry about that, right? But gas gangrene, I'm sorry to tell you, is starting to show up because we have a lot of people who have diabetes. And so with the diabetic condition, unfortunately, because having so much glucose out there in the bloodstream, it starts to destroy the capillaries. And so as the capillaries get destroyed, you'll first see it in like the distal parts of their body, so like very darkened, like lower limbs, okay? And so that's indicative that those capillaries aren't working that well anymore. They're not as oxygenated as they should be. And so those tissues are starting to decay, and you'll see that, for example, their heels will have big fissures, like big, sort of like a cut through the heel, but it's not a cut, it's just kind of split. And so because of the inflammation that comes along with this condition, then a lot of times people wear slides. So now you got the fissure, you got this wearing slides, you know, that's gonna let dirt in. And now these people are, the other thing is if you're not getting good oxygen down there, then your nerves are dying too, and they have neuropathy. They can't feel what's going on. If you have perfringens in your arm or something, you're like, whoa, 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 this hurts so much. You go get some help. They can't even feel that they're having a problem. So it's on you. You see your patient, like, let me just take a look. Let me just see what that looks like. Just confirm if it looks okay. All right, because gangrene is quite serious. And it will absolutely lead to the amputation at that point. Okay, and the last one is difficile. This is C. diff. So people have heard of this. And C. diff is a real problem in healthcare. It forms endospores, almost impossible to destroy. And because it is present in some people, and they'll be put on antibiotics, it undergoes the resting state. Okay, I'm just gonna go in the resting state. And it's fecal oral, okay, so this requires that you're keeping very, very, very good cleaning habits. And if it finds itself on a bed rail, on a door handle, on a wall, on the floor, there's no nitrogen or carbon there, it undergoes sporulation. And so it's waiting to get introduced into a person where it's warm and moist and lots of nitrogen and carbon available. Really hard to get rid of. So people, if they have C. diff, they'll make you wear the max personal protective equipment, absolutely. You'll put it on to go in there to do whatever it is that you need to do, and you can come right back out, take it all off, and you go right into that biohazard bucket, go clean up, and switch over to something else, because you can't take chances with it. Problem is, you know, how do you clean that room? Am I gonna like torch the bed rail till it glows red? Like, that doesn't make sense, right? So it's like, what you can do, it's actually kind of limited. I can't really do a lot of things that I could do against, you know, E. coli, or, you know, we learned about capsules. Oh, capsule. Give me a break. I know how to clean you, and I'm gonna, if I need to. Okay, I have to be aware of it. It's a capsule. I'm going to have to use a particular kind of cleaning agent for a particular amount of time, but I'll get you. because you're vegetative. Endospore, that's a different thing. Yes?
There's actually a company that's making a washing machine for the beds, and it like includes a lot of different chemical processes and heat and everything else. And I think it like takes like an hour per bed to wash.
Easy. Easy. I mean, honestly, you're probably better off like getting a big, huge X-ray chamber. Flip, you know, like, you know, no mess. But I mean, I don't know. I have no idea what these things cost. I'm not just saying that. I'm sure somebody is here like, there's no way we can afford that. Okay, so anyway, bacillus also, anthrax and food poisoning. All right, they'll germinate by bringing in water. That's the main thing, and then enzymes start breaking apart the coat, and then real life begins again for them. Metabolism, they'll become vegetative, and so we deal with them in foods through heating cycles. So let's say I had tomatoes, I bought them at the farmer's market or something. I want to like can them for a while. Then this is what not me, but the company for canning. What they do is they warm up the food, okay, to a pleasant temperature, and then the endospores are like, oh, check it out. There's nitrogen and carbon and moisture, and they germinate. Then they raise the temperature really high. They boil, because that kills vegetative. And then they bring it back down to a pleasant temperature, just in case you missed a few. They let them come out, okay? Let them come out, heat them up again, okay? And then come back down. So it's like a couple rounds. And this depends on how many pounds of material you're talking about. Your table behind this. But that is how you deal with it. Notice that I'm not trying to kill the endospore. I'm trying to trick them. Come on out. Okay, then you blast them. So vegetative are actually pretty easy to kill. Endospores, that's what's tricky. Okay, inside of a person, they're not in the endospore formation. Okay, they will undergo that though if you're on antibiotics against them. Then they're like, oh, I'm just gonna hide. Things get better. Okay, and eventually you'll stop the antibiotic and get it done. So C. diff is really, really problematic. Does anybody have any personal experience with C. diff at work? Of having, okay, so you know what I'm saying. Yes. All right, so you pay special close attention. If they say C. diff, just like avoid that area altogether if you can. Sorry, I missed and you don't have a choice. No. Then just wear the PPE. Wear the PPE, please. All right. So here's the objectives for the prokaryotic cell. And so, bless you. And so we have how and why are they similar, and then dissolved proteins, ions, polysaccharides. Okay. How does it compare to eukaryotes? Well, for eukaryotes, we haven't had our description of this, but you might remember me saying that we have a cytoskeleton in eukaryotic cells. This is absent for prokaryotes. Chromosome, we have one piece, and what shape is it?
Double strand.
I would call that the nucleoid region. Stuff like this is just kind of dumb. Like, okay, I mean, I don't have an electron micrograph for you. So you just have to know they have DNA, it's in there, it's attached at one point at the plasma membrane. All right, how many chromosomes? I already said how many genes per chromosome does E. coli have? We are skipping that. So we have just not had any conversation about DNA, so I just don't want to do that. All right, how do these features compare with eukaryotes? Okay, so we have lots of DNA. I've got 46 pieces of DNA. It's all linear. Okay, I'm diploid, 23 from mom, 23 from dad. All right, why are eukaryotic chromosomes considered to be more complicated? Well, first of all, there's a lot more of it, and they're wrapped around proteins known as histones. Okay, that's to keep it all sort of tucked into the nucleus. All right. Binary fission is a simple process where they simply copy their DNA, okay, and extend the wall and the plasma membrane, and then cut in half. It's super quick. Plasmid, I didn't describe plasmid, so skip 5. 6, the role of the ribosome. Does anybody remember?
Protein synthesis.
Protein synthesis, excellent. How do prokaryotic ribosomes differ from eukaryotics? They're smaller, Okay, how are they similar? They do the same thing, and you know, also two different subunits, small and large. Skipping inclusions, endospores, what are they? They are a resting state. And when do these bacteria make endospores? They will make them when they are out of nitrogen and out of carbon. Okay, so I do say spores a lot, but the real word that we should get used to, I'm including myself in this, is endospore. Simply because spore is also what we term the reproductive process and structure that fungi use. They have spores, right? And there's fungal spores in the air right now, but that's for reproduction. That's not what this is. This is a resting state, so I'm going to try to stick to saying endospore. Okay? And then we know this is about bacteria. This is the resting state that can resist so many things and comes from soil bacteria that are gram-positive bacilli. Yes?
Is there some fungal spores that can cause like similar problems?
No, I mean, there are fungal diseases, absolutely. There's nothing like this. No, we can blast fungal spores. They're nothing for what this is. Okay. Next, these are at the top. Good job. Okay, like all sorts of other things are going to yield way, way, way before them. Okay? All right, so structure and information. We just talked about height, endospores, difference in vegetative. They're a resting state. They're not doing anything. They're just sitting. Okay, so I said, you know, kind of like a seed. Okay, but a seed is for reproduction. But you know, you do have some seeds maybe in your garage right now that you're gonna, you know, have that herb garden and you forgot all about it. They're still good. They're a resting state. Okay? They just need to germinate. They need to be in something moist. Okay? Plant is different. They need sunlight, and it's a little more complicated what's going on there. Endospores don't need that. They don't need sun at all. Okay? So very good. Cell wall. So these are alternating sugars, N-acetylglucosamine, N-acetylgramic acid, alternating to make long rods called glycan rods. Where else in nature is this compound found? No, I'm not asking that question. How do gram-positive and negative bacteria differ with respect to their cell walls, thick and thin? Okay, don't forget the amino acid crosslinks, D amino acids described, they're all labeled. Okay, which eukaryotic cells have cell walls? Oh no, we're not there. We are not on the eukaryotic cell. If you, no, skipping. No, go all the way to the last sentence here. What is the relevance of bacterial cell walls in the treatment of bacterial infections? Okay, first off, your own immune system is going to recognize they're wrong because of the D amino acids, and we're going to have a certain kind of antibiotic that is directly related to stopping the synthesis of the cell wall. All right, flagella. It is, okay, so placement, I can't tell you. It's on the outside part of the cell, okay, but you know, you have one, you have many, I don't know, I can't know this. But I can know that they use it for motility. And today we will see organisms that are flagellated and they're going to dart around, you know, independently one from another. Okay, glycocalyx. Okay, oh sorry, fimbriae and poli. Describe the placement and function. Okay, so fimbriae and poli both are gram-negatives. Structures, and they are there for attachment. They act a lot like Velcro. E. coli in the intestines. Glycocalyx. So what is the glycocalyx? It is a sugar coat. It is composed of polysaccharides and is outside of the cell wall. And so capsules and slime layers. You know, I'm gonna just, I wish I could alter this. Just draw a line through that. I already told you the difference between them, and it's really not worth asking you about it because you can't see the difference. I can't see the difference. Okay, the capsule is held more closely, the slime layer is more loose, but I can't see either one. But biofilm is something I want you to know about. Okay? Biofilm is a major problem in healthcare. It starts on a non-living surface, something that's been introduced inside of your patient, and it doesn't need to be forever like a stent. It could be the IV. Okay? So an IV, for example, let it change every once in a while. Because what if something's wrong on there? Right? We've just got to recognize that. Okay? Functions of capsules and biofilms. Okay, this is going to protect the cells within from phagocytic cells, antibiotics, a lot of cleaning agents. the protective layer. All right. Was there something beyond that? I think there is. I have that down. This is right. Good. Okay. Yes.
Quick question for clarification. For biofilm, is that something with limitation or can it be like on the table surface or?
Well, yes. I mean, strictly speaking, yes, but I'm not worried about the table
Oh,
surface.
yeah. I just wanted to know if it could.
Oh, yes. In fact, one of the pictures that I showed you was of basically a sewage pipe, you know? Okay. We're skipping on here. All right, but now, list the functions of the plasma membrane that are exclusive to bacteria, so you should be able to do that. Give me one.
Homeostasis.
Is that exclusive to them?
DNA attachment.
DNA attachment, that's right. So homeostasis is absolutely a key feature of plasma membranes, but that would be true for anybody. Okay, so that would be both in my example. All right, label shapes and arrangements. So hopefully you can do this. What is this? It's single coxy, diplocoxy, staphylocoxy, streptocoxy, exactly. I could have put a diplobacilli, I don't know why I didn't. Okay, so this is Vibrio and spiral and spirochete, and these would all be spiral though, if I just asked for the shape. Okay, so now here, this is a gram positive and a gram negative, and I know because I see the wall represented here and here, and it's much thicker over here. Boy, they're being really generous, this is 4 layers. Okay, so you're just never gonna get it. You just have to know. Okay, so what do I want to say? This is a gram positive, this is a gram negative. This is the plasma membrane. Okay. This is the cell wall right here. Okay, this is cell wall or peptidoglycan right there. Okay, I don't know what I'm trying to show here. If it were one of these, it might be the lipoic acid, but that's not it. Okay, so this is the outer, so most places will say that this is their cell wall. I don't like doing that, because you're gonna get this mixed up. Let's call this the cell wall, and I want this structure here to have its own name. What do I want? The second phospholipid bilayer. Outer membrane, excellent. And here we have the porin and LPS. Now, that's often really hard to show. It would be, see these little teeny tiny lines coming up? That's the old polysaccharide. So I'll see what I can do to make it more evident to us. And one thing here, there's fluid layer here, below the outer membrane, and on the other side of the wall as well. We call that the periplasm, and it's full of hydrolytic enzymes. Yes?
Do they both have that?
They both do.
Gram positive and gram negative.
Have what?
The periplasmic space.
Well, there are some enzymes here in the gram positive that act similarly, but the majority are gonna be on the gram negative. So it's almost always like an afterthought for gram positives. Okay.
Gram negative is gonna be the only one with the second,
Yes,
positive.
yes, yes. Yep, and today we'll do the gram stain, and then we'll see that the whole thing is based on this. Are you a gram positive or gram negative? And we'll see it's key to a bunch of thoughts that we'll have going forward. Okay, parts of the source is the DNA. So then I think I wanted this to be the cortex. Okay, and this is the exospore, sporium or something. I don't know what else this may be.
Wait,
Oh,
I'm
the
sorry.
The inner plasma membrane, yeah. Yeah. All right, endospore, sorry. Yes. So it's typical for me to have pictures. Okay. And I get them all from the internet.
Wait, I'm sorry, did you just go over the endospore, cause I couldn't hear a thing?
Oh, okay, yes, so this is the DNA right here. See that?
Yep.
Okay, that's the DNA. Okay. This is the cortex. Okay, and this is like the exospore. And I said I don't know what I wanted here, maybe that's why I took the arrow off.
Okay.
It would be one of the 2 plasma membranes that's here and here. Okay. All right, endospores are resistant to many things that would otherwise kill vegetative cells. So this is actually what we're going to talk about next unit. List a few of the things that endospores can resist. So they can resist boiling, they can resist UV, they can resist most chemicals. Okay. be very successful at killing normal bacteria, normal vegetative bacteria, not gonna work. Alright, what makes an endospore so resistant? It is the fibrous proteins and the DPA protecting the DNA. And the fact that they don't have any water, that's part of sporulation, to pump the water out. So they can't be boiled. That doesn't work on them. Okay, now I want to make something very clear. If we think about where are most organisms capable of sporulation found? The soil. So if we take the time to get rid of the dirt, and that means sweep, dust, like that, I'm not afraid anymore. Now I can just use my normal cleaning stuff. Okay? But if I haven't done that, and there is dirt there, then that's where my concern is. Okay, so I know no one likes to dust, but that's it. Alright, so that is that part. Now we have to keep moving. Okay, so now we're on eukaryotes. Nope, have to download it. I'm sorry for this. Goodness, I did get it here earlier. Okay, so with eukaryotic cells, you know, in some semesters I have sort of skipped it, like, oh, I didn't have time, and like just keep moving. But the problem is that when we get to the immune system, it's going to be entirely about the function of some of our white blood cells, the macrophages, T cells, B cells, and how they function. So if we don't kind of lay the foundation right now, that becomes very difficult.